Well, welcome everyone to our January Dunlevie lectureship and Happy New Year. I hope everybody got a little bit of downtime and ready to plunge into a new year. So just a few center updates regarding the spring 2026 seed grants. Applications for the seed grant program is going to be open soon. The center will fund two awards, $35,000 each for 12 months, starting July 2026. And full program details will be posted at our website. Our next center lecture is going to be on February the 12th, and we are honored to have Dr. Mana Parast. She is a professor of pathology and the director of perinatal pathology at the University of California, San Diego, and she will be talking to us on research into the human placenta, challenges and opportunities. For today, I'm handing things over to Dr. Virginia Nguyen, who will introduce our very own honored speaker, Dr. Yoel Sadovsky, and he will be sticking to the theme of placenta. So it's all yours, Virginia.
Great. All right. Well, Happy New Year, everyone. It's my pleasure to introduce Yoel Sadovsky. He is a recent addition to Stanford. He comes from having had a long and prestigious career. He ran the McGee Women's Research Institute from 2007 to 2024. He has served as the president of SRI and the Perinatal Research Society, also been elected to the National Academy of Medicine, Association of American Physicians, and the Royal College of Obstetrician and Gynecologists, as well as AGOS. And he was awarded the Cozzarelli Prize in Biomedical Sciences from the National Academy of Sciences in 2013. His work expands across many domains of understanding human placenta, from looking at RNA transcription decades ago to now looking at non-coding RNAs, extracellular vesicles, as well as looking at processes of oxidative stress, ferroptosis, as well as lipid metabolism within the placenta. So I look forward to hearing about fat placentas, and so I'll turn it over to Yoel.
Thank you very much, Virginia. Can you hear me okay?
Yes.
Thank you very much. It's really great to be here. Great to be sitting in one of the small telephone rooms here at the CAM, and talking about science is always uplifting to me, hopefully to all of us. Yasser, I really thank you very much for giving me the opportunity to speak. I'm so impressed with the Dunlevie activities, from clinical activities, clinical deployment of healthcare, and down to research and translational research and basic research. I think it's fantastic. So as Virginia said, I'm a perinatologist, MD, but I made a choice in recent years to really focus my entire academic activity on understanding early human development, understanding fetal growth. In that context, of course, the placenta plays a key role. So I've been studying the placenta, and our research in our group has really focused on everything from very, very basic biology of gene regulation, very nitty-gritty molecular analysis of placental cells, but all the way to the clinical application, translational research, and the clinical application of that research. So over the time, we've done some work that I'm not going to talk about today necessarily on understanding gene function and small RNAs and all kinds of things that I know are maybe of interest to many of us. Today, for this talk, I chose for a brief presentation on some translational aspects of our research focusing on placental fat. So why do we study placental fat? So I really want to try to understand it. It's going to be a brief talk, but I want to try to highlight why the placenta gets fat, why is it important for the placenta to get fat, where's the fat coming from, how does it get into the placenta, and where it goes, and what can go wrong when there's too much fat or enough fat, and why it's important. I will say right now that many of us are talking about something called lipotoxicity, which means toxicity related to fat. And it's a bit of a vague term. It's something that we all use on labor and delivery. We say reactive oxygen species and oxidative stress and lipotoxicity. But what does it really mean, especially in the context of placental injury and in the context of delivery of fat to the fetus? What does it really mean is something I'd like to shed light in the next 20, 25 minutes. So I'm going to share my screen. I actually find it fun to talk without the slides, but I will share my slides. Can everybody see my slides? Let me just do the presenter.
Yep, we see them, and they're now in full screen. All good to go. Thank
you very much. So I like photography, and I just love, as I said before, I love the California oak. So I just, every once in a while when I can, I just add some pictorial depiction of my habits into the slide. So why does the placenta get fat? So let's put it in a broader context. Our major interest is not just in fat. Our major interest, is in what really happens to the embryo from early development. So you can see, can you see my arrow? Okay, what happens from the early developmental stages of human development? So here, this is the embryo, the blastocyst, prior to implantation, all the way to mature pregnancy. So the timeline for this arrow is 9 months of pregnancy, hopefully 9, hopefully not less than 9. And we think that in every normal pregnancy, across the 9 months, there's an ongoing balance between injuries that are depicted here in red and adaptation to injuries by different mechanisms depicted here in green. So the example of injuries, I think the most common one in the Western world is hypoxia and hypoperfusion of the uterus leads to hypoxic stress. There's all kinds of other types of stress. Infection, inflammation is very important. Environmental stress and malnutrition, very important in some parts of the world, even the US. Genetic and epigenetic changes. And of course, also, unfortunately, social determinants of health are very important, although we don't always understand why. In our laboratory, we're interested not only in this normal process and the injuries that occur, but also in the innate inherent mechanism depicted in green that are designed to protect against those injuries. So example, I just put some names here, microRNAs, non-coding RNAs, different proteins, metabolites, extracellular vesicles that communicate some of the protective mechanisms, and they're all leading to adaptive mechanisms. And we think that the balance between the injuries shown in red here and the defense mechanism shown in green here, that balance hopefully leads to intact pregnancy after 9 months. And if something goes wrong, such as that the red influences supersede the green protective mechanisms, that's when you start having diseases. Examples would be preeclampsia, example would be fetal growth restriction. And here on the right bottom side, I put a picture of twins that I delivered with one healthy placenta attached to a healthy baby and an IUGR fetal growth restriction baby attached to this placenta. And of course, this was a sick baby. And that's exactly the situation that our lab and others in the field have been interested in understanding what really is going on here, which is really the fundamental phenomenon underlying the significance of my talk today. So just to make sure we're on the same page, here is a cartoon of the placenta from Williams Obstetrics, and we all, I think, remember the fetus is connected here, the mother is here, and the human placenta is a villus placenta, meaning that the fetal blood comes in villi, goes back to the fetus through the villi, and maternal blood fills the spaces in between the villi and goes back, comes from the mother side, goes back to the mother side. And if you look very closely, this is really what I want to focus on today. If you look very closely, if you look cross-section in one of those villi, that's what you see up here. So you see the mother's blood would be out here, fetal blood would be here, and this is a picture of what we call terminal villus. where the only thing that separates the mother's blood here and the fetal blood here is a layer of trophoblast cells, which are fused to form what's called syncytialized cells or multinucleated trophoblast cells, and they're shown here in light blue. Every once in a while, there's the mononucleated progenitor cell, cytotrophoblast. And in the villus core, there's also some macrophages, fibroblasts, and so forth, endothelial cell. But really, the key exchange between the mother and the fetus occurs right here. This area is called vascular syncytial membrane. That's the physiological term. And it's very similar to the lung, except for in the lung, the interface is between air and blood. And here in the placenta, it's interface between maternal blood and fetal blood, so blood-to-blood interface. And what really separates the mother from the fetus is the trophoblast extension here, a basement membrane, and endothelial cells. And it is those trophoblast cells that are of fetal origin that are regulating all of the key transport functions that occur between the mother here and the fetus here. Oxygen exchange, CO2 release back to the mother, exchange of nutrients, waste products, production of hormones, also mechanical defense. All of these occur right here at the level of trophoblast. So these are the cells we focus on, and I just want to summarize the key unique features of the human placenta. It is discoid, looks like a disc, very much like a mouse placenta, which is very different because human placenta is villus, mouse placenta is labyrinthine, and I'm not going to get into this. I will show a picture or two just in a little bit. The human placenta is what's called hemochorial, which means that the trophoblast cells that you see here in light blue are directly bathed in maternal blood. That's what hemochorial means. And it's also chorioallantoic, means that the connection of the placenta to the fetus is through the allantois, which transformed into the umbilical cord, as opposed to through the yolk sac, which is called choriovitelline. So why is placental fat related to fetal growth restriction? I think all of us know the growth curves of the placenta and the fetus across human pregnancy. So the x-axis is weeks of pregnancy, the x-axis, the y-axis is weight. And we all know that the placenta makes a very rapid growth in the beginning and then kind of flattens and grows very gradually, whereas the fetus initially grows very slowly, but then it really accelerates after 20 weeks. And if you make a parallel from that growth pattern to what actually happens as far as the accretion of fat in the fetal placental unit, it happens that happens that in the middle of pregnancy, about half of the caloric accretion in the placenta and the fetus comes from fat, which is represented by the yellow here in this bar. But if you look at as pregnancy progresses, and if you get close to term, the majority of the actual calories that get accrued in the placenta and the fetus, their source is fat. So obviously fat somehow has to come from the mother and also potentially produced in the placenta and get into the fetus to allow this growth. And I think all of us in clinical perinatology and obstetrics know that babies who have fetal growth restrictions, such as shown in this slide, have a decreased amount of fat accumulated in different tissues. And that's one of the ultrasonic approaches to measure fat. You can see here abdominal fat mass is decreasing FGR, subscapular fat mass, decrease in FGR, mid-arm fat mass. Many of these parameters, not all of them were significant in these studies, but many of them have been significant, showing a decrease in fat accumulation in the fetus when the clinical condition of FGR develops. So this actually led our lab and others to be interested in the metabolic fuels that cross the placenta into the fetus. And of course, glycogen and glucose are very important. Amino acids, of course, are very important. We focus on many of these, but I want to focus mainly on lipids right now because we think, and hopefully others in the placental field would agree, that that has been a relatively understudied area in placental biology. So how do things work? This is another electron microscopy picture of the same interface that I showed you in a cartoon. Here's maternal blood would be here. Here's one maternal red blood cell. Here's the fetal red blood cells. And this is the interface that mediates the trafficking, in our case, of fat from the mother into the fetus. So how do we think it's working, inferring from other fields where there's fat transport in other tissues? So here's maternal blood in this cartoon here in the bottom. There's maternal blood. will cross the microvillus membrane here, which is right here. So that's MVM, the trophoblast microvillus membrane. It will get into the cells that are depicted here in light yellow. Some of the fat will stay in the cells, and I'll show you in a minute what it looks like. And some of this will get broken down to free fatty acids and cross into the fetal capillaries and then into the fetal compartment. That's how the fat will cross. So and I'll get into this in more detail in just 1 minute. So our basic premise has been that probably in conditions where there's inadequate trafficking of fat into the fetus, something is wrong in this area, that the trophoblast does not uptake enough fat. So if this is true, if the trophoblast does not uptake enough fat, what we'll see in cases of IUGR, for example, fetal growth restriction, FGR, is that we'll see a decreased amount of fat accumulating in the placenta. That was our initial hypothesis, and we're wrong. So I'll show you in a second what actually happens, but we're wrong. It was not about the actual uptake of fat into the placenta. So here's what should normally happen. Normally, triglycerides on the maternal side should be they're too big to be uptaken by trophoblast. So they need to be broken down by lipases at the surface to free fatty acid. Free fatty acids get uptaken into trophoblast by specific transporters. And then once they're inside, some of the fat will transfer directly to the fetus, which is on this side. Some of the fat will serve as signaling molecules that can regulate gene expression in the nucleus. And some of the fat will get accumulated in lipid droplets within the placenta. And we thought, incorrectly, but we thought that the problem in fetal growth restriction would be here, that there will not be enough uptake of fat into the placenta, and that's why there's inadequate trafficking of fat and inadequate fat accretion in the fetal placental unit. So we did those studies in cultured cells and also in mice. And just to mention here that I'm not going to talk about cholesterol uptake. It's another mechanism, which is complicated, but it's not really relevant to my talk here. When we took cells and exposed them to a common injury that occurs that leads to FGR, such as decreased oxygen supply and hypoxia. So you can see it here. So standard conditions of growing cells versus hypoxia, or in biopsies from women who had either a normal pregnancy or FGR pregnancy, in both cases, in vitro and in vivo, in primary human trophoblast cells, we saw that in hypoxia or in hypoperfusion led to fetal growth restriction, we saw increased accumulation of fat in the placenta, counter to what we initially thought. We thought there was going to be decreased uptake, but we saw more, and you can see it quite clearly here. This is the old-fashioned medical school oil redox staining. You can see it here. Also here, you can see staining in trophoblast as opposed to normal. And here, this is a fluorescent stain for lipids, and you can see this very colorful yellow signal depicting fat in the placenta in case of hypoxia. And this is just a time course of the same phenomenon, another type of staining, but you can see very nicely lipid droplet accumulated in hypoxia in the placenta, counter to what we originally thought. Okay, so that's kind of interesting. How does it actually work? So there's 4 major types of lipids that we need to assess if they traffic or not traffic, what happens to them. Triglycerides, cholesterol, phospholipids, and non-esterified fatty acids. So the studies here on the left are in vitro studies. The studies on the right are in vivo studies, meaning in fetal growth restriction placentas. So this is biases from human. That's why I emphasize the translational impact of our studies. This is from real patients who had FGR baby, and this is from cells in primary human trophoblast in culture. And if we look at the 4 major types of lipids, in each case, you can see here labeled in star, or here that I put a red marking around it, it was mainly triglycerides that accumulated in trophoblast, not cholesterol, very small changes in cholesterol, phospholipids, and non-esterified fatty acids. Mainly triglycerides got accumulated in the injured placenta, either in vivo, in FGR, or in hypoxia in vitro, hypoxic conditions. So if this is so, and basically in vivo in injury, and in vitro in hypoxia, which is a common injury in the Western world, if mainly triglycerides get accumulated, how does it actually work? And we came up with 4 potential mechanisms that may lead to accumulation of triglycerides. One of them is increased uptake. Maybe the placenta just uptakes more triglyceride, therefore there's more lipids in the placenta. A second potential mechanism is increased synthesis within the placenta. A third one was decreased oxidation in the placenta that would lead to increased amount of lipids in the placenta. Well, that was a no-brainer because if there's hypoxia, and basically to break down lipids, you need a process called beta oxidation. Well, if there's hypoxia, we expected to see decreased oxidation, decreased breakdown. And what we didn't know about is efflux. How much of the fat from these cells get transferred into the placenta? And I'm not gonna, because of time, I'm not gonna get into all the studies, but just to summarize a lot of work, we found that there's no change in the uptake, absolutely no change in uptake, absolutely no change in synthesis, but we expected decreased oxidation. That was not surprising. But what was surprising to us was decreased efflux. For some reason, the trophoblast cells in injury in vivo or hypoxic injury in vitro did not release enough fat into the fetal side. They tend to retain the fat, and that's why we saw more lipid droplets. So how does it work? Well, lipid droplets, here's a cartoon, and here's a real electron microscopy for lipid droplets. How does it work? Lipid droplets have proteins on their surface, and you can see here an example. The yellow is lipids, fat is yellow, and on the surface they have phospholipids. You can see them in blue, and proteins on the surface. And those proteins tend to regulate the accumulation of lipids or the breakdown, what's called lipolysis. And there's families of those lipid droplet proteins called plin proteins. These are the lipid droplet proteins or associated proteins. And we didn't know which one of them are relevant to the placenta. So again, cut a long story short, we found out that in the placenta, only 2 members of this family of proteins that regulate the lipid accumulation in lipid droplets, only 2 proteins are expressed. One of them is called plin2. This is the old name, adipofilin. Another one is called plin3. This is the old name, TIP47. So is it possible these 2 proteins are relevant to not only they're expressed in the placenta, but they also regulate the accumulation of lipids in the placenta? So we did some studies, and I'm just going to abbreviate those studies. We published them several years ago. We found in condition of hypoxia, not only there's more lipid droplets, but also there's a massive increase in this protein called plin2. There's absolutely no change in plin3. So of the 5-6 family members, only 1 protein called plin2 is upregulated in case of hypoxia, and the other protein we found in the placenta did not change, did not do anything. So obviously we decided to study this protein a little bit. And again, I'm just gonna give you the results of lengthy studies done by Ibrahim Biltirici, who was a very talented postdoc in our lab. And we found that we knock down this protein plin2, the cells start dying, basically. As if this upregulation of lipid or lipid accumulation protects the cells against some injury. If we overexpress this protein that we identified, if we overexpress it, it improves cell viability. Again, consistent with the function of this protein in protecting against this accumulation of lipids, irregular or abnormal accumulation of lipids in the placenta. Plin3, when we manipulated this protein, there was absolutely no effect. So I just want to summarize what I told you so far. What I showed you that in injury in a placenta, in vitro or in vivo, that lead in vivo leading to IUGR, of course, we know that the consequences could be fetal growth restriction, so in interference with fetal growth. And we also know that lipids can potentially cause what's called lipotoxicity, lipoxic damage to cells. And that's commonly being discussed, but really an ill-defined entity that I'll come back to in a few minutes. And what we also showed is that the same proteins that we found to be upregulated, plin2, they actually protect the cells against the injury, such that here is the injury, but as I said before, we're focusing on the adaptive responses to injury. Plin2, this protein, helps the formation of lipid droplets, and it helps to contain the lipids such that they would be less damaging to the cells, because too much lipids, as I'll show you in a minute, is bad, and I'll show you why. So if for nothing else, I'm going to try to shed a little bit more light in my remaining time on why lipids could be damaging. We know they're important for fetal growth. We know they're critical for fetal growth, but they could be damaging, and I'll show you why. So how does it work? The dynamics of the lipid droplets obviously becomes critical. If all the proteins just contain lipid droplets, okay, that's protective for the cells, but we still find injury. How does it actually work? So I don't want to get into all the details of the biochemistry of how breakdown of lipids happens and all the enzymes, not really critical for us. I just want to focus on the lipid droplet for the next few minutes. So let's focus on lipid droplets. Here's the trophoblast, again, fetal blood here, maternal blood here, hopefully well-oriented. And at the lipid droplet, I already showed you this purple protein, plin 2 specifically, which is important for the lipid accumulation to protect the cells against the injury. Now, the opposite from accumulation is lipolysis. And lipolysis is regulated in many tissues by specific lipases. Many of us in medical school learn about hormone-sensitive lipase, HSL. I can tell you this protein is hardly expressed in the placenta. Barely you can detect it even by PCR, so I don't think it's really relevant. And then there's a cofactor that was identified about 10 years ago, named CGI58, not important the name, that is essential for the function of the lipase. So just to recap that point, plin is a protein that helps the formation of lipid droplets, and the lipase and the cofactor helps with lipolysis, meaning the breakdown of the lipids from the lipid droplets. So here we got a balance between a protein that helps the formation of lipid droplets and proteins, enzymes that help the breakdown of lipid droplets. So we decided to focus on these proteins to understand what happens, why the placenta actually accumulates fat, and what happens when there's breakdown of the fat. So I just made a little cartoon here. Here's the plin protein, here's the cofactor, here's the lipase, 3 players. So again, just to recap, and that work was done by Jennifer Guerrero, another very talented postdoc that helped us to decipher those pathways. When you have lipogenesis, meaning the formation of lipid droplets, the plin protein that helps lipid droplet formation becomes part of the lipid droplet, and the lipase that breaks down lipids is actually not relevant here, not attached to the lipid droplet, but the cofactor is attached to the lipid droplet membrane, but there's no lipolysis. Basically, all the lipids get accumulated in the droplets. Where there's the opposite process, the lipolysis, this protein, plin 2, gets detached, and the lipase with its cofactor get bound by lipid droplets, and then they mediate the lipolysis process. So we thought that this may be actually a way for us to understand why there is accumulation of lipids and what happens with this lipolysis. So I'll just go very briefly over the studies. We knocked down, either in vitro or in mice, both the lipase or its cofactor. And just to abbreviate a lot of work, when we knock down the lipase, hardly anything happens. Here's a mouse placenta with a knockout, very similar to wild type. But when we knock out the cofactor, which is labeled here in red, you can see the accumulation of lipid droplets in the placenta, massive accumulation. When you knock down both proteins, no difference from the CGI58 knockout, meaning that the cofactor deficiency led to accumulation of lipids in the placenta. This is studies in vivo also, but also we have in vitro, where we have showing nicely the accumulation of lipids in the placenta by either measurement of the total lipids or the area of the lipids in the mouse placenta, both showing us that it is the cofactor that you see here in red, the cofactor, which is the key protein that is essential for fat accumulation. If you don't have this protein, you don't have lipolysis. So, right, when I get excited, I start moving the computer. Sorry.
So Yoel, quick question.
Yeah.
So if knocking out the lipase didn't change accumulation, is there another lipase that is?
It's great thought. It's an excellent question, excellent thought. And that's exactly what I'm going to show you.
Oh, okay.
You know, when you give a talk and you get questions, you have 2 responses. Sometimes you say, the speaker says, oh, this is an excellent question. That means that I have an answer for it. Sometimes you say, oh, that's a really interesting question, meaning I have no clue what you're saying. So this was a great question because I do have the answer. And just to recap, also, when we when we have deficiency of this CGI58, the cofactor, and we add it back into the placenta, we get restoration of the normal amount of lipids. So again, suggesting to us that indeed we're on the right track. So as Virginia just asked, yes, we thought, okay, there must be another lipase that regulates this activity. It couldn't be just that the cofactor is doing the whole thing, because the lipase deficiency is bad for you. And indeed, again, cutting a long story short, we did identify there's another family from this family of lipases. This is the one that we thought was important, but was not that important. But there's another one called PNPLA9, which is very important for this fat accumulation. And just to show you, when we knock it out, you see lipid accumulation. You can see it in mice in vivo. When we knock it out, we see much great increase in lipid droplets. And that showed us that this protein PNPLA9 is important for lipid accumulation. So essentially what I showed you so far is that this protein PLIN2 is important for lipid accumulation, lipid droplets, and this lipase, not this one, but this one is important for the breakdown, and it functions with the CGI58 with the cofactor. And it's mainly the cofactor, which is the major one that leads to lipid lipolysis in the placenta. Now, in the last few minutes, I wanna tell you what the things that I just told you, how are they important in the context of what's called lipotoxicity? That many of us is using this name, the reactive oxygen species, lipotoxicity on labor and delivery. We talk about injuries, blah, blah, but what does it really mean? So this is my last few slides. What we found was very interesting to us several years ago is that the mice that are deficient in this cofactor, not only they accumulate lipids, but all of a sudden we saw that they accumulate much longer types of fatty acids. And this is a mass spectrometry that shows the length of the fatty acids that are bound by triglycerides. And you can see here, I think all of us, irrespective of the kind of academic activity we do, all of us can see that the longer fatty acids get accumulated in this knockout. Now, this is really important because many of us know that some of the long-chain fatty acids, especially long-chain polyunsaturated fatty acids, such as docosahexaenoic acid, DHA, or arachidonic acid, docosahexaenoic acid, they are really critical for brain development. They're really critical for fetal development, eye development, and so forth. But surprisingly, in the knockout, not only we get more lipids, but also more of the long-chain polyunsaturated fatty acids are so important for development got accumulated in the placenta. So this is really not good because the fatty acids that are most needed are the ones that now get accumulated in the mouse placenta that lacks this cofactor. Now, here is why it's important. We and others discovered a process called ferroptosis. And I just want to tell you what it is and why it's important for lipotoxicity in the placenta. Ferroptosis is, as the name implies, it's an iron-dependent form of cell death. Now, it's very similar to something called apoptosis, programmed cell death, very similar to something called pyroptosis, which is an inflammation-related cell death. Ferroptosis is an iron-dependent cell death. And it happens when you have iron, which is capable of donating electrons in a Fenton reaction, many of us may remember from medical school, and when you have oxygen and something called oxidizable fatty acids. So this is just my cartoon of a phospholipids that has two fatty acids and a phosphorylyl ethanolamine in this case, that here, for example, may contain a fatty acids, and the fatty acids that has multiple double bonds like DHA, like arachidonic acid, they are the ones that are most susceptible to attack by hydroxyl group like hydroperoxyl or hydrogen peroxide or other hydrogen reactive oxygen species that can cause damage to these molecules. So the longer polyunsaturated fatty acids, the longer they are, the more double bonds they have, the more susceptible they are to attack by what's called these nucleophiles that may potentially attack them and cause hydroperoxidation and causing the formation of what's called reactive oxygen species, what we call in clinical medicine. This process leads to cell death by a process called ferroptosis. I just want to show you here a cartoon of the membrane. Again, not essential for us to all the step, but here's a membrane. Here's a membrane that is damaged. And here in the middle of the membrane, any cell membrane, you have those phospholipids, And when they get peroxidated, like with this addition of OOH, they get damaged. And that leads to this process called ferroptosis and tissue injury, which is really an underlying process that we call commonly lipotoxicity, reactive oxygen species accumulation, oxidative stress. Many of us talk about this in the context of preeclampsia. So when we analyze for these specific species that are hydroperoxidated, you can see it here. We found in biopsies of placentas from preterm birth, there's a higher accumulation of those hydroperoxidated, highly oxygenated free fatty acids in preterm birth. This is not cells in culture. This is real biopsies from women who had preterm birth. So this became really interesting for us. Not only, I told you how fats can get contained lipid droplets, but if you have excessive amount of fat or the lipolysis takes over, you get release of those long-chain fatty acids that can really damage the cells. Let me show you a little cartoon what cell damage looks like. So look at this. These are cultured cells exposed to ferroptosis. So take a look at this. Here you see the cells. They look nice. And all of a sudden, you're going to see like explosion of cells and formation of those membranes. See the cells like literally explode. I hope you like this movie because I just love this movie. So I'm going to play it again. Oops, sorry. I just want to play it again because I just like it. Oops, now I can play it again. Okay, here it is again. Look at the cells. They look fine and normal. And we expose them to ferroptosis. Look what happens when they start exploding. You see this cell here. Look here. Look at these cells. You saw that? And now they form those membranes. Those membranes are representative of the cell membrane getting exposed to higher osmotic pressure. They cannot withstand it, and they form those membranes, then they explode, basically. So how does it really work? Well, I'm not going to get into all the details, but just in one or two slides summarize that we think that this other lipase that we identified is very important, protecting the cells against lipolysis that can lead to the release of those dangerous fatty acids from triglycerides. And when you cross mice, for example, that are heterozygotes, and you form mice that don't have this enzyme, you see the knockout and you apply ferroptosis, the much greater amount of level of embryo death, and you can see one example here of embryo death in those pregnancies. So altogether, we think that this process of ferroptosis is critical for preterm birth and for preeclampsia, and I don't know about FGR at this point, we haven't studied yet. But just to focus on our findings here, which is highlighted in the orange bracket, we identified this process of ferroptosis that leads to placental injury, just like injury in other organs. We found also that the enzyme that we identify in the placenta protects against this injury by reducing the amount of free fatty acids that can get hydroperoxidated and lead to ferroptosis. And we do think that this process is critical for preeclampsia. In fact, so much so that in a paper we just published, you can see it here in the bottom, the hypertension, we just found that there's some certain drugs that recently have been implicated in protection against ferroptosis, against preeclampsia, such as the proton pump inhibitors that were identified. We found that they specifically block this process and help to attenuate the process of ferroptosis in placental cells. So just to wrap up, our discoveries in the context of placental transfer trafficking of fat, when fat comes from maternal blood, gets broken down the surface, gets into trophoblast, and then gets released into the fetus, we identified a abnormal mechanism where in the case of injury, such as hypoxia, in vitro or in vivo, there's much greater accumulation of lipid droplets, which is fine, which is a protective mechanism, and we identified the protein mediates it. We also found mechanisms by lipases, but most important by the cofactor, this cofactor CGI58, that are critical for breakdown of, and when there is breakdown, there's much higher likelihood, especially during reperfusion, when you reintroduce the oxygen, much higher risk of the process called ferroptosis, and we found in spontaneous preterm birth in the placenta and also in preeclampsia. So this is our contribution to this specific field. Again, I just love the scenery in our beautiful Bay Area. And these are the people who contributed to the work directly over the years. And these are people who work in our lab on other projects. And these are some of our collaborators, and the studies were supported by the March of Dimes and by the NIH. And I'm going to stop here, and I guess we do have a few minutes for questions. And thank you very much.
Great. Thank you so much, Yoel. Beautiful summary of a lot of work. Quick question. So just to clarify for myself, when you're inducing ferroptosis in the system, you're not adjusting iron, you're adjusting lipid injury and adding basically hydrogen peroxide. Is that correct?
That's very correct. Yeah, it's absolutely correct. And the reason why we're not adjusting iron, other people did adjust for iron. They increased the amount of iron, you see more ferroptosis. We did not do it because the placenta is loaded with iron. It's one of the tissues that has a very high amount of iron as it is. So maybe it's not surprising that in the right conditions, it is prone to this form of cell death called ferroptosis. So you got the placenta is really loaded with the three major components that are needed for ferroptosis. You got iron, you got oxygen, and you got free oxidizable fatty acids. Oxidizable meaning they are the long-chain polyunsaturated fatty acids. And these very fatty acids that are so essential for fetal development, they are also the risky ones, like DHA, like arachidonic acid, and related ones.
So conceptually, is the term oxidative stress that we talk about with hypoperfusion and the oxidative stress that leads to preeclampsia, these are all the same process in a way. So whether it's ferroptosis or oxidative stress, we're talking about the same process?
Yes, we're talking about the same process. I'm not going to say that ferroptosis is the only mechanism leading to oxidative stress, but it's probably the major one. And I do want to emphasize, Virginia, something that many of us tend to forget. This is not a chronic hypoxia situation. Chronic hypoxia will not lead to ferroptosis because if you have chronic hypoxia, you don't have enough oxygen. And ferroptosis depends on oxygen. But it is what's commonly happening in obstetrics. Hypoxia reperfusion. We all say it on labor and delivery. We talk to our residents. We talk about hypoxia reperfusion with uterine contractions, with umbilical cord compression, with other processes. Hypoxia reperfusion. It is the reperfusion process when you reoxygenate an injured tissue that leads to this ferroptotic injury.
Okay. Great. Any other questions? I'm looking in chat. I don't see.
Yeah, Matteo has a question.
Okay.
Hi, Yoel. Fantastic talk. A quick question. Is there a way with your mouse genetics that you can induce the phenotype only, for example, in the placenta versus the fetal side and see the contribution it may have? And also another question I wanted to ask, because I saw the blebbing that happens when you induce the ferroptosis. Is it known what happens to the cortical cytoskeleton of the cells, like this blebbing? There is like fourth generation actomyosin contractility. Do you know anything or what's happening there?
Yeah, jokingly, I said before how speakers respond to questions. So your first question was excellent. The second question was interesting, but I have no clue what the answer is.
It's okay. No, just a curiosity.
No. Great question. But for the first question, yes. So we can generate this phenotype in a mouse only using lentiviruses that target either gene knockout or overexpression specifically in the placenta. We have ways to do it. I didn't want to get into the technology, but Matteo would be glad to talk about it. But using lentiviruses at the blastocyst stage, we can specifically either knock out or overexpression gene in a blastocyst, and then the phenotype happens only in the placenta, not in the embryo. And I have a lot of data for that. Your second question, no, we don't really know what happened to the cytoskeleton, like actin and other components of the cytoskeleton. We do know that membranes, the phospholipids are damaged. And I have some other studies actually we published where we did some other molecular modeling of the damage, what actually happens when the phospholipids, especially long-chain polyunsaturated fatty acids, get damaged. And we do have models for that, but I didn't want to get into the brief time. But Matteo, I'll be glad to share with you some of our data and what we plan to do. I plan to continue some of this research here, so hopefully we can continue about this.
Yeah.
Fantastic.
Actually, in thinking about the videos again, I had a sense that the cell actually did a huge contraction before the explosion.
Yeah, yeah, you're right, because there's absolute dysregulation of the regulation of the osmotic pressure inside the cells. And you're right, they contracted, then they exploded, but before they exploded, they started forming those blibs, and then they exploded. And it's very dramatic. That's why I show this video.
All right. Well, I think we are at time. Thank you again, Yoel, for a thought-provoking talk, and look forward to interacting in the future.
Thanks a lot. Thanks for the opportunity. Thanks
Great
for
talk, Yoel. Thank you.
Thank you. Bye-bye.